All right, let's work through another example here. So for this problem, our participants are not people, they're actually rats. So in this study, the researchers injected rats with this drug, midazolam. I'm not actually sure what it is. I think it's a sedative. And so rats were randomly assigned to either a control condition with no injection, a same condition where they got the same amount of a dose that they were used to, or a different condition where they got a different amount of the dose that they were used to. And then we looked at their ambulatory behavior, so basically their movement, I think through a maze over the course of 6 trials. So if we look at our data, we have each rat is assigned to one of these groups, and there's 8 rats per group. And each of those rats provides 6 trials, 6 observations. The numbers in the actual dependent variable is their amount of movement, counted in terms of, I think, seconds of movement during the trial. So if we look at our data here, we can see that we have 3 groups, the different group, the same group, and the control group, and 6 observations per rat. First thing we do, of course, is fill in our degrees of freedom. So down here for our convenience, I have our degrees of freedom. Notice here, I have t minus 1. I've switched that to k. Actually, it should be k minus 1. Remember, k is our number of repeated measurements. I had it as t here to mean time, because in this case it is time, but recall that repeated measures aren't always time, so it's better to label this k. That's what I'll do from here on. This is just a little holdover. It's a little mistake from an earlier version of the slides. But so I have my degrees of freedom done. The next thing I can do is compute my sums of squares total, which again, I won't spend much time here. It's just each score deviation from the grand mean squared and take the sum. So I can fill that in to my source table. Next, I will complete my between subjects sums of squares. To do so, I need to first compute each subject's mean. So this is the first rat's mean trial times across all 6 trials was 88.3. The next rat was 211.5, and so on. Subtract each subject mean, x bar s. Subtract the grand mean from the subject mean. Square the result, multiplied by k, the number of observations, right? 1, 2, 3, 4, 5, 6, because that's how many observations made up this mean. Finally, I can take the sum to find my between subjects sum of squared deviations. Recall that if I had my total sums of squares and this much belongs to the between subjects, whatever's left over is my within subject sums of squares. So within subjects is just total minus between. Next, I think it's helpful to be able to work not from the raw data, but from a summary table like this. If we had just a summary table of the means of each cells, we can do the rest of this, we can solve the rest of this problem. But it's important to understand how to read a table like this, a summary table like this. So for example, how many observations made up that mean? Well, and the answer is n, lowercase n, the number of rats that were in one of these conditions. So in this case, it was 8. There were 8 rats in each group. Likewise, how many observations made up this marginal mean? Right, where this is the mean of all the time for data. Well, it would be 8, 8, 8. It would be n times g, right? The number of rats per cell times how many groups that we have. So in that case, it is 24. We had 8 rats times 3 groups. Likewise, how many observations in this marginal mean? And the answer is 48. 8 rats measured 6 times. So that'll be important moving forward as we start to compute our sums of squares. We need to know how to weight those sums of squares. They need to be weighted by the number of observations. Finally, how many observations total, right? And that would be n times k times g. So 8 rats times 6 observations times 3 groups. 144 in this case. Likewise, because our sample sizes are all equal in this case, we can compute the marginal means right here. So this marginal mean is just the mean of the 3 cells. In that case, it's 286.21. And likewise, this marginal mean is just the mean of these 6 cells across. So if we were given just these means in the cells, we can compute everything that is in these colored cells, and we would know how many observations made up each mean. So we could solve this problem just from this summary data, just from the summary means table. So we've done our between subject sums of squares, our within subject sums of squares. So now we need to start partitioning that variance further. So we'll start by partitioning the between subject sums of squares into group and error. And to do that, we need to compute our group sums of squares. So each group's mean, x bar g, minus the grand mean squared, times the number of observations that make up that group mean. In this case, n times k. So for example, the control group mean is 130.88, right? That's the mean of all the observations for the rats in the control group. We can subtract that from our grand mean of 169.02, square the result, and multiply by how many observations make up that mean. Well, 8, 8, 8, 8, 8, 8, n times k. In this case, 8 times 6. We can do the same thing for the same condition and the same thing for the different condition. Take the sum of all 3, and that is our group sums of squares. Remember, at the between subjects level, we only have condition or group and error. So whatever is not condition is left over, and that's error. So to find the sums of squares error, it's simply the sums of squares between subjects minus the sums of squares group or condition. Coming back to our means table, we can now find the sums of squares for our repeated factor, which in this case is time, where we take the mean of each time period, so time 1, time 2, time 3, x bar k, subtract the grand mean, square it, multiply it by the number of observations that make up that mean. So n times g, 8, 8, 8. I had 8 rats times 3 groups. So for time 1, I have my mean of 286.21 minus the grand mean of 169.02 squared times 8 rats times 3 groups. And I can do that for time 2, time 3, time 4, all the way to time 6. Taking the sum, I get the sums of squared time. Next, I need to do my interaction term, but remember I can't go directly to that. The first step is to compute my sums of squared cells. So here I'm just taking the mean of each cell, subtracting the grand mean, and multiplying by the number of rats, in this case, in each cell. So that's 8. So for the first cell, that would be the control condition time 1. That's this first cell here. I have 213.88 minus the grand mean of 169.02. Square that times 8. I can do the same thing for the control time 2 cell, 93.25 minus 169.02 squared times 8. And I can do that all the way for all my cells until I get to the diff times 6 cell, 178.63, minus the grand mean squared times n. The sum of that gives me my sums of squares cells, but recall that my interaction term requires me to subtract the main effects sums of squares from sums of squares cells. So my sums of squares for my interaction term is my sums of squares cells minus my sums of squares condition or group, same thing, condition, group, minus sums of squares time, the repeated measures factor. So in this case, that is shown down here. And error is what is left over, right? I had 761,755.84 sums of squares at the within subjects level. I simply subtract out the time and the interaction term to be left with the error. Moving forward, I compute my mean squares the same way I've done all along, dividing my sums of squares column by my degrees of freedom column. And I compute my F's. Remember, my F at the between subjects, I'm using my between subjects error. So it's the quotient of these two cells, 142,907.52 divided by 18,320.1. Whereas at my within subjects level, I'm using my within subjects error term as the denominator. And I guess I didn't present the F table, but these are all significant effects. So I have significant main effect of my condition, my between subjects condition, a significant main effect of time, but that main effect is qualified by a time by condition interaction. So the effect of time depends on the condition that you're in. Conversely, the effect of the condition depends on which time period we're looking at. And we can see that if we go back to our plot. Right? The effect of time is different in this square dashed line different condition as compared to the control condition. There's clearly a different effect of time. Likewise, there's a different effect of condition depending on which time period I'm looking at. At time 4, they all look pretty much the same, but at time 1, they're all fairly spread out. So that's what an interaction is. The effect of one independent variable is dependent on the level of the other. So I'm not going to go through all of the post hoc tests again here, but I'll go through the between subjects one to see just again why it's important to do it by hand, and we can compare it to the SPSS version. So if I want to do my between subjects simple effects, my between subjects follow-up test, what I could do is just go compare means. I could come to my one-way ANOVA and put each time period in my dependent variable list. So this is essentially saying do a one-way ANOVA at each time period. It's kind of another way of splitting the file. It's just doing it separately for each time period. And when I do that, it will give me the one-way ANOVA at each time period. But again, see that it is giving me a different error term, which SPSS calls within groups, but this is just our error term, right? It's giving me a different error term at each time period. And it's only giving me 21 degrees of freedom because when it computes the ANOVA on time 1, it's not using any of the information from the other time periods. So even though I have that assumption of homogeneity of variance, and I could have a better estimate of population variance by using all of the data, when I split the file, the computer's not doing that. Instead, the computer is treating it as if it only has time 1, and then as if it only has time 2, and so on and so forth. So instead, a better way for doing between subjects post hoc test or simple effects is to use our hybrid mean squared error to combine parts of the between and within subjects model to compute the mean squared error and degrees of freedom. And remember, we can just plug and chug these numbers to find our mean squared error and our degrees of freedom. But I said, recall, that I would recommend making an Excel spreadsheet to do this. So that's what I did. I just made an Excel spreadsheet where I can type these numbers in, and then it'll give me the answer. So I defined the equation up here before, and I double checked it against a result that I knew was right. And so every time I need to go and compute the mean squared error and degrees of freedom, I can do that easily because I just did it one time and then I let the computer do the heavy lifting for me, instead of computing these numbers each time by hand, which is prone to just basic errors, not arithmetic, but just transcription errors, and just, you know what I mean. We make mistakes when we have to do things by hand. So let the computer do the heavy lifting for you. You do the thinking, but let the computer crunch the numbers. Looking again at my ANOVA table, if I replace, then I can let the computer compute the between subject sums of squares, because remember, that won't change. But I can replace the degrees of freedom and the mean squared error with the hybrid version that I computed here by hand. And that will give me more stable estimates. I have more degrees of freedom, so I have more power to observe effects, and this mean squared error isn't going to bounce around, right? The fact that I get 6,388 here and then 7,863 here, that's not ideal, right? Because I'm assuming that these were all drawn from populations that have equal variance. So the fact that these variances are different it's probably just sampling error, right? I don't want to use all these different variances. Instead, I want to use one overall one using all the data. And by doing that, I get more degrees of freedom. And in this case, we can see that for 2 and 57 degrees of freedom and an F critical of 3.18, and I recompute my Fs here. So I recomputed each F. I can see that one of my results actually changes. Whereas at time 5, this between subjects effect was significant when I ran it by splitting the file. It's actually non-significant when I use the hybrid mean squared error, when I use all the data. I would say that this T5 here, what SPSS is calling a significant effect, is actually a type 1 error. When I use the more stable estimate, I find that it's non-significant. So by using that by hand, I actually protected myself against committing a type 1 error. So again, to reiterate, I can split the file and have the computer compute these sums of squares for me to do the heavy lifting, but then I should replace the degrees of freedom and mean squared error with the ones that I computed by hand using the omnibus test. And recall, if we were doing within subjects tests, we wouldn't have to even compute these. We could just take the mean squared error directly from the omnibus test. So that's a lot simpler. That's why I'm only showing you the between subjects here, because it's a little bit more complicated. All right, well, that is it for mixed ANOVA, and that is it for the content of the course. So congrats for getting to the end.